switch. But anyway, okay, let's start because it's 11. And good morning, everyone. I really like to welcome to our second Indoor Ag Science Cafe. Let me start recording before I forget. Well, actually, it looks like it's recording.
I think you started recording already, but
yeah, okay. I think it's recording. Okay, good. So, quickly, just to remind, this is the sort of community to discuss about the technologies going into indoor agriculture, indoor farming, specifically for the what we call sometimes vertical farming. And then we have two parts. One is a sort of topical presentation, which sometimes goes to a little bit scientific. And then second part is more like an information exchange, asking questions for you guys, for us, or exchange information, learning for us to running something going on for us as a scientist, you know, so that's more like an information session. And then this first part is recorded, but the second part is not recorded. So hopefully you feel more comfortable to ask questions or share the issue, to heads up for the other members in this group. So we have three more Indoor Ag Science Cafe. And today is Erik Runkle as a presenter. So I'm gonna stop sharing here and then let Erik take care of. Stop share. Okay, should be. All right.
okay. Where did that go, Cherry?
Yeah, so the share in the center, if you put the cursor down in your screen, you have a green.
Oh, of course.
Yeah, share, and then select the, yep.
Okay, very good. Okay, well, welcome everyone. As Cherry said, if there are questions that pop up, please go ahead and unmute yourself and interject. And there's no such thing as a dumb question. So it's always hard now to gauge, what level people are. Some are very new, some are quite experienced. And so certainly there might be some things that are not clear. So please ask questions as we go. And so I chose to talk about lighting, and lighting, of course, is a very broad topic. We often think of lighting in 3 different ways, or sometimes even 4. We think of it in terms of intensity, in terms of the quality or the spectrum of the light, the photo period, and sometimes we're even now concerned about lighting uniformity. So how it's distributed over a 2-dimensional or even sometimes 3-dimensional area. And so before I start, it's important to acknowledge most of this research has been generated by my graduate students. Yujin recently finished her PhD. She's now a postdoc with me. And Qingwu Meng is a PhD candidate expected to finish at the end of the year. Also want to thank a lot of lighting companies and granting agencies that have supported this work. And this is, I would say, the major ones that have contributed to our research the last 6 or 8 years or so in this project. So I want to talk a little bit about what far-red radiation is and some of the different considerations with far-red in terms of light perception from a plant perspective as well as the human. So by definition, we usually refer to far-red as 700 to 800 nanometers. Occasionally you see a different definition, but that's generally the accepted broadband between 100 nanometer broadband. So relative to the other photosynthetic photons, which is typically 400 to 700, the photons have less energy. So a couple things. One is that far-red is just outside our perception of light, or we can perceive it, but it appears very dim to us, very, very dim to us. And so a lot of far-red LEDs have a peak emission of around 735. There will be some photons from that get close to 700 or maybe 710, and we might perceive that. If it's a fairly bright irradiance of far red, we might see it as a kind of a deep magenta color, but generally we don't see it very well. This is the McCree curve, which refers to the relative quantum efficiency of photons at eliciting photosynthesis. This is a curve that was generated in the early 1970s and is often used to describe instantaneous photosynthesis. The point I want to make here is that when you measure when you measure instantaneous photosynthesis, you can see that blue and green and red are all relatively effective. The most effective photons are typically the orangish color. Looking at far red, you can see that the photons decrease quite quickly. And so once you get to about the 735 nanometer peak, on an instantaneous basis, those are considered to be poorly effective at stimulating photosynthesis. Now, far red radiation is perceived by primarily the phytochrome family of photoreceptors. So there are different photoreceptors or pigments in plants that perceive light. And so the major photoreceptor that perceives and acts upon far red radiation is phytochrome. There are two forms of phytochrome. There's a red absorbing form, PR, and that has a peak absorption of around 660 nanometers. And then there's a second form of phytochrome, that's the far red absorbing form, and that peak absorption is around 730, 735 nanometers. So what's nice actually with LEDs is that the peak emission of one of the, probably arguably the major red LED is 660. That corresponds well with peak absorption of the red absorbing form of phytochrome. And likewise, 735 peaks well with the peak absorption of the far red absorbing form of phytochrome. And so phytochrome is never in one form or the other. You can see that there's overlap, and so basically this pool of phytochrome is in some sort of equilibrium between these two forms. So what we're trying to do is basically looking at far red radiation. Is what you're doing is when you're delivering it, you're altering that phytochrome pool in an attempt to elicit some sort of plant response. So very simply speaking, these are some of the major events that phytochrome mediates in plants. The germination process of seeds and deetylation. Phytochrome mediates what we call shade avoidance responses. So if plants are growing under a canopy, the environment is rich in red compared to far red. And in response, plants tend to survive under those conditions. And so responses include stem elongation, leaf expansion, as well as petiole elongation. We typically see a suppression of branching, so we have more apical dominance or more growing of the primary shoot tip. And in some cases, we can get early flowering as a way for the plant to try to survive flower and complete the reproductive process. We also know that phytochrome mediates flowering of some crops, and that's especially the case with plants that flower faster or require long days for flowering. Beginning in about 2010, I was part of a project with Cherry Kubota, Carrie Mitchell, and several others who are still in our current team. It was a 5-year proposal focused on LEDs in horticulture, and our thrust was to look at how different wave bands of radiation and how they interacted to influence flowering of various floriculture crops. To boil down a lot of research into one slide, what we have here is the effect of different light colors delivered as night interruption lighting during a 15-hour dark period, so a 15-hour night. So this is a petunia that has a fairly strong response to long days. If we grow plants on the far left, you can see that they are vegetative and not in flower. If we give incandescent lighting for 4 hours during the middle of the night, you can see that the petunia flowered. Incandescent lamps emit a lot of red and far red. If we gave plants instead red plus white LEDs, so there was no far red in the spectrum, you can see the plants are vegetative. And yet when we gave the red, white, and far red LEDs combined, you can see that the plants are in flower. And so basically this tells us, and a lot of other research shows and confirms, that for some of these crops, the inclusion of far red promotes flowering. So Eugen Park has done a lot of work the last couple years investigating more the plant responses to far red, and her focus has been on ornamental crops. So I'll show a handful of slides showing far red responses to ornamentals, and then switch over to some examples with leafy greens. So here we have seedlings that are grown indoors. The only light source is under these LEDs. In this case, all of the plants received blue light at 32 micromoles per square meter in second. And then you can see these snapdragons were exposed to different intensities in micromoles from either red radiation, which is R, or far red radiation, which is FR. So on the far left, we have 128 micromoles of red with, of course, the 32 micromoles of blue. There's no far red. And these next two treatments, we are partially replacing some of the red with far red. So in this case here, we have equal amounts of red and far red. And the PPFD, or the photosynthetic photon flux density, is remaining constant. The next three treatments are keeping the red and keeping the red and blue constant, just adding incrementally more intensities or greater intensities of far red. So what we see, and as what we would expect, is that when you add far red or you partially substitute some of the red with far red, you get a promotion of elongation, you get taller stems, and you also get typically larger leaves. I could also point out that you typically also get a little bit of a paler color green leaf. These leaves are a little bit more of a yellow-green compared to the environment without far red. And so we see that is the case whether we are substituting some of the red with far red, or when you begin to add increasing amounts of far red, you get taller plants. Now, what we're interested in this case with floriculture propagules is there a residual or a carryover effect of these environments on the subsequent flowering response? Because of course we're growing these plants specifically for them to flower. So after transplant, these were then grown in a greenhouse in a common area, and data on time to flowering and flower number were recorded. So here is then the plant response after transplant in the greenhouse. On the far left, there's no far red. You can see that they flowered an average of 31 days at 68 degrees Fahrenheit after transplant. The other 5 treatments all include some far red, and you can see that when that far red was included during the seedling stage, that accelerated flowering by about 10 to 12 days. So clearly for these types of crops, if rapid flowering is desired, there's a value in including far red in the radiation spectrum. Now, there are lots of nuances with far red, and we oftentimes, with far red and other wave bands, we should not be thinking of these wave bands in isolation because they do interact with each other. And so that's been a focus of Eugen, is to look at how red and far red interact with other wave bands, or in this case, interact with the photosynthetic photon flux density, or PPFD. So here we have on the far left, these 3 treatments all would be considered a low light environment. So we have 96 micromoles per square meter per second delivered for 18 hours a day without far red, or with 32 or 64 micromoles. And then these 3 treatments have 4 times the amount of light. So the red to far, there are 32 micromoles of blue in all treatments. You can see here we have a dramatic increase in the amount of red light. With that, we're keeping the red to far ratios the same. So the ratio of red to far red is 2 or 1, and likewise the ratio is 2 or 1. This is geranium, another common ornamental crop. You can see again in the absence of far red, you typically get smaller leaves with short petioles, and that's also the case under high light without far red. And as you add that far red, you can see greater elongation. And what this study shows is that the elongation is typically greater when you have a low PPFD than when you have a high PPFD. And so that is consistent with some other research that has looked at what triggers the shade avoidance response. One way is by delivering low intensity of light, and the other way is by adding far red to the radiation spectrum. And so you can see that the contribution of far red and the effects on elongation growth depend upon how much photosynthetic light is also delivered. So those plants were then grown again in a greenhouse after transplant. And this was a bit of an unexpected result because geranium is a day neutral plant. We would have not have predicted an effect of far red in promoting flowering. You can see time to flower here is not influenced by the addition of far red. But interestingly, when we added far red to the highlight environment, we got promotion of flowering by 5 or 6 days. And so we have some theories to try to explain that, but again, it was a bit unexpected. So one other part I want to talk about is ratio of red to far red and how that interacts with blue. We know that blue light also regulates and can inhibit extension growth. And so we wanted to look at how the red to far red ratio interacted with blue to influence growth of different crops. So this is coleus. It's a shade-tolerant bedding plant or annual. Here you can see grown without any blue and only red. You can see a relatively tall coleus as you would expect. And as you add increasing micromoles of far red, you get a progressively taller plant. What was interesting though is that then when you have 80 micromoles of blue, which is a relatively high intensity, you saw a relatively strong suppression of extension growth, and then the addition of far red to these intensities, you can see had minimal effect at promoting flowering. And so what we're finding is that a high intensity of blue light can at least partly, if not completely, override the effect of the red to far red ratio. And then just another example, in this case with geranium again, you add far red, you get progressively longer petioles, and that continues to be the case. In this case, you still get a red to far red response, but it is diminished when you include blue in the spectrum. Okay, so let's shift here and talk a little bit about lettuce. So this is some of the early work that William Meng or Qingwu Meng has done. Just exploring the different interactions of blue, red, and far red. And so in many cases, you might see this sort of spectrum or this sort of ratio where you primarily have red photons, but then a moderate amount of blue. And you can grow a decent quality lettuce. And so then the question is, what if you then add 30 micromoles of far red? What if you don't have the blue and only red? Without far red, but equal amounts of blue and red. And you can see these other treatments here. And so what's quite compelling, and again, it's not necessarily novel, but we want to quantify these things about how the radiation spectrum influences the different growth attributes. And so we're looking at extension growth, we're looking at leaf size and leaf length, fresh weight, dry weight, leaf color, potentially leaf texture, potentially leaf taste. And so there are lots of different attributes we can look at, and even at some point we'll get more and more into the phytonutrient content. So there's certainly, as with floriculture crops, a lot of similarities. You add far red, you get extension growth, but in some cases that may not be desirable, especially when you don't have blue. In other cases, the inclusion of some blue with some red, with some far red, may end up giving you the sort of leaf morphology you're looking for. And so we can then capture these plant growth responses. These were grown just for a short period, but the greatest dry weight, in this case that's what we measured here, was highest under the red-blue combination with the addition of far red, and the lowest was that same red-blue without any far red. In fact, we had 30% lower dry weight when we didn't include far red in the spectrum. So this is on the cultivar Rex. We've also done the same work on Cherokee. This is a red leaf variety. Of course, it's only red if the light environment or some other factor will bring out that purple pigmentation. It's fairly well known that blue light as well as UV will bring out that purple coloration of the leaf. And likewise, when we don't have blue and have far red in the spectrum, you tend to get the palest color leaf, which is what we've already seen with floriculture crops. So then you begin to get some trade-offs where If you want the purple pigmentation, you might get it with this treatment, but this was also the treatment that had the lowest fresh or dry weight. Okay, so starting about 3 years ago or 2.5 years ago, we became limited in terms of number of treatments we could deliver at one point in time, and the amount of area we could grow plants in. And so I developed the Controlled Environment Lighting Laboratory, which is here at Michigan State. It was completed early last year, and we've been doing research since spring of last year. And so this is a video that William took just showing some of the lighting fixtures that we have in there. These lighting fixtures were developed by Osram Innovation. They're meant to be research modules where we can independently control 7 different wavebands of radiation, ranging from UVA all the way into the far red regions. So there are narrowband wavebands, UVA, blue, green, red, and far red. There's a mint white, which is basically a greenish white LED, as well as a more of a traditional warm white. And so we've been using this facility since then, and it's a facility that we plan to use in this project when this project gets funded. So I just want to show one experiment that we've completed that William has done in CELL. So CELL is the acronym for Controlled Environment Lighting Laboratory. In this case, we had 10 different treatments. And in these conditions, you know, they're highly controlled environments so that we are not potentially influencing the effect of the radiation spectrum with another parameter. So we can be confident that temperature is the same, CO2 is the same, and any treatment results are because of the treatments of the spectrum. So we had a photosynthetic photon flux density of 180 micromoles in each case. In this case, we have what we would consider maybe a somewhat traditional spectrum of 120 micromoles of red with 60 micromoles of blue. And then in 3 treatments, the blue was at least partly or fully replaced with green. Here, the blue was partly or fully replaced by far red. So actually, in this case, the photosynthetic photon flux density was reduced compared to the other treatments. And then we have 3 other treatments. This is with replacing some of the blue with green and far red. And then as basically 2 controls, we have a warm white, which you can see the breakdown of the different wavebands, has a fair amount of green and red. And then the mint white, which again has about 60% of the photons are in the green waveband. So lettuce was grown under these treatments. And this is just a photo of a representative plant from each of those treatments. And as you can see, some strikingly different morphologies as well as colors of the different lettuces. And so, you know, I don't think there is such a thing as an optimum spectrum. I think then the question becomes, what are the sort of characteristics that you want? And then we can elicit those characteristics to some extent based upon manipulating the light spectrum. Now, I will mention that these are static lighting treatments, meaning that the spectrum does not change. And one of the things we are proposing to do is to grow plants under a spectrum for a particular time, and then maybe transition the plants to another spectrum to elicit some other desired growth traits. So these are just photographs, and I'll show some data here in a minute. So this is for the green leaf variety Rex, and then for the red leaf variety Rochei, or I've heard lots of different ways that that's pronounced. But you can see, again, big differences in coloration. That's probably most striking, but also in leaf expansion. And I think if those of you are listening and look at the pictures, you can probably imagine that if you pull a leaf from each of these plants, they're going to have some different characteristics. The textures will be different. Some people might want a thinner texture of a leaf. Some might want more of a crunchy leaf. And so what we want to try to do is learn more about how these different wavebands interact to affect these different dimensions. And the challenge is the interaction, and that we really can't study them in isolation. So I've got 2 graphs here just showing some of the data. So plant diameter was one metric to measure basically the area of the plant. Not surprisingly, it was greatest when we had the red and far red. And then as as you increase the blue relative to that, you get progressively shorter plants, or I should say narrower plants. If you replace some of the blue with green, you got a little bit of a greater leaf area. And then you can see that actually the mint white and the warm white, the plants grew quite well, and you have all the different radiation wave bands represented in those treatments. So that's diameter, and then here is the total dry weight. And so you have the biggest plant in terms of radiation capture, and maybe not surprisingly, then you have the greatest dry weight and probably fresh weight as well. But you also don't have that purple color that you may want. And so again, that ties well into things that we want to do, where you might grow these plants under a spectrum to increase radiation capture, to try to increase the weight of the plants, and then maybe later on you switch to a different spectrum to try to get more of the pigmentation and coloration. So with that, that's the end of this presentation, and I'd be happy to take any questions or items that you want to discuss in more detail.
Okay, let me, thank you, Eric, very much. Let me stop recording here.